What if I told you that the exact laboratory technique that helped cure some of humanity's deadliest diseases also blinded scientists to an entire hidden world? In nature, microorganisms don't live alone. They thrive in massive, chaotic populations made up of countless different species. But to actually figure out what a specific microbe does, scientists first had to figure out how to isolate it completely. Wow. This concept, known as a pure culture, simply means growing a mass of cells of just one single species in a laboratory vessel like a test tube. The wild journey to achieve this started in 1878 with Joseph Lister, who managed to get the first pure cultures using a mind-bending technique of serial dilutions in liquid media. Using a specially constructed syringe, Lister painstakingly diluted a fluid, which was likely milk, containing a mixture of bacteria, until just one single organism was delivered into a container of sterile milk. After incubation, the bacteria in that container were all identical to the parent cell, and he proudly named this isolated organism Bacterium lactis. But the real game changer was Robert Koch, who took these methods to a whole new level by smearing bacteria on glass slides and adding specific dyes so individual cells could finally be seen clearly under a microscope. Koch didn't stop there. He revolutionized the lab by adding gelatin and agar to create liquefiable, solid culture media, allowing him to grow isolated growths called colonies, where millions of individual bacterial cells were packed tightly together and could easily be transferred to other media. Using these painstakingly precise techniques, Koch studied material from patients with pulmonary tuberculosis, applied his rigid series of tests just as he had with the anthrax bacillus, and shocked the world by discovering the exact microorganism that causes TB. The impact of pure cultures was monumental, allowing scientists to isolate the exact microbes responsible for fatal infections, vital soil nitrogen fixation, and specific fermentations. But here is the massive plot twist. This strict obsession with pure culture techniques and Koch's postulates sometimes led brilliant investigators down absolute dead-end streets. Because they were only looking at isolated cells, early scientists completely missed the existence of viruses, and they had absolutely no idea that two or more microorganisms often cooperate to cause a complex disease or even bring about a desirable fermentation, like the complex ripening process of your favorite cheese. Today, science has evolved, and we realize that to truly understand biology, we are just as interested in the chaotic, mixed microbial populations and their combined effects as we are in pure cultures. The true future of marine microbiology, rumen microbiology, and decoding the deep secrets of our own intestinal tract will depend entirely on first understanding the physiology of individual microbes, and then decoding the complex ecological relationships of total microbial populations thriving together in a given environment. If you are ready to dive deeper into the unseen microscopic forces that actually run our planet and our bodies, hit that subscribe button for Microbio.